I will be discussing the phenotype Parkinson's disease and the gene SNCA today. So what exactly is Parkinson's disease? Parkinson's disease is a neurological disorder that gets progressively worse. It specifically impacts the central nervous system and dopamine-producing neurons. Dopamine is a neurotransmitter, which means that it's a chemical messenger. The dopamine-producing neurons in the substantia nigra die, and there seems to be an accumulation of Lewy bodies and alpha-synuclein within the same region. So in this picture right over here, the blue area is the substantia nigra. So the dopamine-producing neurons die there, and the Lewy bodies and alpha-synuclein seems to accumulate there too. Researchers believe that understanding why this occurs could be vital in understanding more about this disease. Symptoms do start to appear slowly, and the most prominent ones are tremors, which are most distinct at rest, muscle rigidity, and slow voluntary movement. Also, every case of Parkinson's is different, which is another reason for why researchers are still figuring out the cause and the cure for it. Parkinson's disease is the second most common neurodegenerative disorder. It affects about 1 million people over 50 in the United States and 4 million people worldwide. It also affects 13 people per 100,000 and more males than females. 85% of diagnoses actually occur in people with no family history of Parkinson's. Next, we're going to be talking about the etiology of the disease. The heritability is 0.362. That means that the environment plays a bigger role than genetics do in the variation. The shared variance, c², is also very small. All of this data comes from twin studies that were done from Sweden and only looked at males age 65 and over. Those twin studies are from 2 separate papers. GWAS studies showed 525 association results and 39 genes. One gene that showed up frequently in the association results was SNCA, which I'll discuss further later. Its p-value was 4.000 times 10 to the negative 73. MAPT, another common gene which functions in DNA binding, had a p-value of 2.00 times 10 to the negative 48. Very early on in this project, I did a simple Google search to see what the most common genes are for Parkinson's, and LRRK2, which functions in ATP binding, was the first gene to pop up. It was also on GWAS and had a p-value of 1.000 times 10 to the negative 39. Now let's talk about the gene, SNCA. SNCA is on chromosome 4, right over here. SNCA codes for the presynaptic chaperone protein alpha-synuclein found in the brain. Let's break that down. Presynaptic means that the protein is found in a neuron before the synapse, and the chaperone protein is one that helps in the folding of other proteins. So, alpha-synuclein is a helper protein found in the presynaptic neuron. This gene, SNCA, is also associated with Alzheimer's disease and alcoholism. Low levels of alpha-synuclein can be protective against oxidative stress, while high levels can lead to neurodegenerative disorders, such as Parkinson's disease. There is a hypothesis that researchers have come up with stating that low levels increase cravings for alcohol, and as alcohol consumption increases, SNCA expression increases, which can lead to neurodegenerative diseases. When looking at the gene ontology, SNCA has 10 biological processes, which includes antigen processing and presentation of endogenous peptide antigen, 2 cellular components, which includes MHC class II receptor activity, and 13 molecular functions, such as MHC class II protein complex. All in all, SNC has a lot of different kinds of functions. SNCA has 6 different exons, most of which are relatively small. Exon 6 is the longest at 2,562 base pairs. After mRNA processing, the gene is 3,042 base pairs in length. Before mRNA processing, the gene is 137,970 base pairs. The picture that you have in front of you is the full canonical gene product, which comes in at 140 amino acids. There are 188 orthologs present in 280 species for SNCA, but the most similar transcript to humans are in primates, such as the bonobo, chimpanzee, gorilla, and orangutans, which have a 100% target ID and an 85.05% query ID. That means that they have 100% similarity to the human gene, while humans have an 88.05% similarity to primates. When it comes to paralogs, there are only two of them, SNCB and SNCG. For both of these genes, the target percent ID is higher than the query percent ID. So that means that the vertebrates have a similar gene to humans, but the human gene is not as similar to the vertebrates. This graph has sequenced RNA within normal tissues on the x-axis and reads per kilobase million on the y-axis. According to this graph, SNCA is most highly expressed in the brain and second highly expressed in the bone marrow. This graph, which has the same axes as the previous one, also indicates that SNCA has the highest expression in the brain, whether it's the whole brain, the cerebellum, or the fetal brain. The next highest expression is in the liver. This graph is a little different. We are actually looking at SNCA gene expression. On the y-axis, we have transcripts per million, and on the x-axis, we still have various different tissues that have been sampled. The most highly expressed location is still the brain, and even though we are looking at all of these different types of tissues, we can see that the most highly expressed region within the body is the brain. Next comes the tibial nerve and whole blood. All in all, it seems like SNCA is most highly expressed within the brain. It doesn't look like natural selection has been acting in the immediate vicinity of this gene because none of these signals have crossed either one of the lines that would indicate that a selection event took place, whether it was recent or ancient, complete or incomplete. When it comes to polymorphisms, SNCA has 852 variants with a minor allele frequency or an MAF larger than 0.01 and would affect the protein sequence. One such polymorphism was RS77141123, which had a nucleotide change from a cytosine to an adenine and an amino acid change from an aspartic acid to a tyrosine. Aspartic acid has an R group that is a carboxylic acid and is an acidic amino acid, while tyrosine has an R group that is a phenyl, so it's just hydrophilic and it is a much larger R group than aspartic acid. The minor allele frequency is 0.043 for the variant, and in its highest population, it was actually 0.16. So, all in all, this variant is deleterious and probably damaging. In the map in front of you, you can see that Africa has the highest allele frequency for adenine, while East Asia populations have the smallest allelic frequency for adenine.